Children.
Well, thank you very much. It's a great pleasure to come and learn from jENS and also to be part of this very exciting pharmacology session. So I'm going to talk about clinical trials in general and then illustrate how one of the initiatives can help you as it develops over the next 4 or 5 years. So when I was young, doing clinical trials for medicines was easy. All you had to do was have an idea, write a protocol, do the study, publish your paper, get promoted, live happily ever after. Does everyone recognise that model? Is that how we want to do science? Well, maybe not, and things have changed. And now when we do a study, I'd like to suggest that we have to work in a different way. And we have to build from existing knowledge and work with family advocates. We can't do anything without talking to families and their advocates. And we need, rather than write a paper that gets us promoted, we should look for impact that changes clinical practice through prescribing decisions, through better dosage regimens, through marketing authorizations, through national guidelines. And to get to that impact, we really need to start off with the existing knowledge, and from the existing knowledge work out what are the most important gaps, and then have the idea. I think the genius having the idea who then drives everything else, those days are gone, because the genius needs to fit in with what everybody else has done already and use that information to design the study and conduct the study as effectively as possible. And then once you've got your ideas, then you need to discuss it with everyone who can help you do the study. I'd estimate that if there's a doctor in charge of the research, the doctor needs to do about 5% of the work, and the nurses and the pharmacists and the regulatory managers need to do the other 95%, the statisticians, the trial managers. And you won't get anything done unless you talk to all these different people. So modern science involves teams, involves building an alliance that allows you to do effective science. Then after extensive discussion, you end up with a concept, and then the concept gets adjusted to different funding mechanisms, which is often people's goal. And then once you've got the funding, then you need to do the hard work of designing the protocol, and then conducting the study, and then disseminating it. And disseminating it is what you're all going to do today, which is fantastic, but it's also about papers, and it's also about making sure that people who write national guidelines see your work, and then you get the impact. And the different discussions to have, we need to use existing knowledge as much as possible, so that we're only doing work that needs to be done. We're not reproducing work unnecessarily. Sometimes we have to reproduce work to test and refine, but sometimes we have to look for the gaps and focus on some gaps. And that existing knowledge needs to come into the discussion, needs to come into the concepts that we're trying to develop. You won't get the funding unless you describe how you relate to existing knowledge and the protocol and so on. And then by the time you get to dissemination, then you're feeding back into existing knowledge and changing existing knowledge with your work. And the impact is making sure everybody's heard about the fantastic new knowledge that you've got. And again, this has to be done with the families at every step. The ideas have to work for the families, they have to be acceptable, they have to meet family needs, and discussions and the concepts and so on have to be influenced by the families, who will then play a big role in dissemination and impact. And that's why it's so valuable to have EFC and I here co-chairing jENS, so that we have some lessons on how to do the dissemination and the impact. So at this point, you're all saying, why can't we do Model 1? Why can't it be simple? How on earth am I supposed to do all this? I'm a busy fellow, I'm a busy pharmacist in training. How on earth am I supposed to do all this work and keep all these balls in the air? How do I juggle everything? And the answer is this team science, that we have to work together, that it's not about geniuses by themselves, it's about teams working together so that we all can benefit and we can all deliver this vision where we know what's missing and we fill that gap. So some of the gaps and existing knowledge can be matched with techniques such as extrapolation or modelling and simulation. So this is a reliable, objective, transparent way of using existing knowledge to work out which dose to give, to work out how to assess the medicine, so that we're not guessing about dosages. We're not having to study every medicine in every child every time. We can sometimes scale down from bigger children before coming into neonates. And we can do modelling and simulation about the pharmacokinetics. We can do modelling and simulation about the trial itself, clinical trial simulation, to work out if we change this inclusion criterion, how much longer will the trial take. And all of these factors can help us build in the existing knowledge. There are many gaps. Personally, I think the biggest gaps that we have in neonatal drug development relate to how to measure things and what's the natural history of the conditions we're trying to treat, and how do we segment the conditions into different areas. So pain, for example, we give lots of babies lots of toxic medicines in the hopes of treating their pain. Lots of clever people spend lots of time working out the best dose of morphine or fentanyl or whatever it is. Personally, I don't understand how to measure pain in premature babies. So if we can't measure pain, it's very difficult to decide what dose to give, to decide whether to ever give painkillers. There are some very effective ways of assessing pain, usually using MR scanners or near-infrared and other things, and we need to make sure those can be used in multiple centres and they can be targeted when they're needed. And then we need other measurements that can be used everywhere. The natural history and segmentation, BPD is a classic example of how we have no effective treatments for this condition because we treat it as one thing. We take a statistical concept, oxygen dependence, and think every medicine will treat oxygen dependence. But oxygen dependence at 36 weeks or 40 weeks could be due to central problems, it could be to airway problems, it could be to small airway problems, it could be to lung parenchyma problems, it could be due to blood vessel problems, pulmonary hypertension. And one medicine is unable to treat all of those. Well, maybe steroids can treat all of those, but we'd be much better off targeting biological mechanisms. And if we look at BPD as a whole, then we're not targeting biological mechanisms. That's how cancer's made efficient strides, that's how rheumatology has made so many strides forward, but neonatology is far behind because we treat medicines to treat statistics. We don't use medicines to treat biological mechanisms. And that means finding out about the natural history, that means finding out about how biomarkers work, and segmenting the disease. So when people stop treating cancer, they've stopped treating leukemia, they've stopped treating B-cell leukemia, they're now talking about hairy cell B leukemia type 22, and they're targeting those very specific mechanisms. And that's where the success and the investment in cancers come from. So for me, these are the big gaps. There are lots of very interesting things to think about in terms of medicines and doses, but unless we get these measurement gaps, these natural history gaps sorted, then we are going to be asking the same questions again and again and again. So people have been studying inhaled nitric oxide in premature babies for many years. Several thousand babies have been randomized to big randomized placebo-controlled trials, and we still don't know whether to give nitric oxide to premature babies, who to give it to, or what dose to start, because people haven't looked at the right gaps before doing the big studies. So, and then impact. We really want to make sure that we're aiming for our impact when we look at the studies. We don't want to design a national guideline study we haven't got the dose right. So think about where your doses are going to be. And the impact needs to inform the ideas discussion. Most importantly, you need to persuade your funders that you're going to have a big impact, otherwise they won't give you the money. Then we also need to think about sites. How many times have we got halfway through the study and decided, well, actually I need 5 sites here, I need 10 sites here? And that's no good, because the other 5 sites want to tell you how to do the study. If you go to them halfway through, then it's not going to fit with their practice, and they can't change and you can't change. So it's much better to involve the sites early on, through bring them in at the discussion stage so they can influence the study, so you can do it quickly by working with multiple sites. So many things to do, and research infrastructures are there to help us do all these different things. So how do I do all of this? I collaborate, I meet nice people at nice conferences, and I then prepare, and then to make things efficient and reproducible and high quality, we should be using infrastructures. And there are several research infrastructures I'm going to tell you about. So I just draw your attention to this concept of preparedness. This paper has just appeared on the website of the European Medicines Agency, and this is a working group between academia and industry and some regulators to think through what can we do to make sure a study is going to work, or what can we do to increase the chances that a study will work. Some people call this feasibility, but everyone calls feasibility something different. So we wanted to bring all the ideas together in this paper. So please read it, and please comment on it. I'm sure it can be improved, so please comment on this. There are two key concepts behind this trial preparedness: that we should take a structured assessment. So not just the problems I've come across, not just the problems my statisticians come across, but look through the literature to work out what are all the problems and how can I address those problems before I start. And the paper does include a list of all the problems that we could find in the literature. And then we also put forward this idea of a concept, a preparedness concept, that is trying to make everything explicit, so you know what your assumptions are, you know where your data has come from, and then you can change things as you learn more. And when it comes to arguing with the regulators, we hope this will be a useful tool so that companies can say, we did all this work, we worked with all these clinicians, and this is our study number, and the regulators may say, okay, you've done it in a systematic way, we'll agree with you. Otherwise, the regulators will say, you don't want to spend any money, you're not looking at enough babies. So we're using this to prevent that discussion. We suggest doing this kind of consort diagram before you start. Work out how many people are available for the study, taking account of the condition, whether you're looking at first-time treatments, whether you're looking at repeat treatments, how many people are available for recruitment, how good the sites are, how many people give consent, and what can go wrong. I wanted to call this the fog of war, but the regulators made up this lovely English word, which I think means fog of war. Things always go wrong. And then you've got enough to work out how to do things. So we want to put this on the table. This is all about making your site is ready by designing the protocol and thinking of the problems before they start. And then these are the sorts of components in the preparedness concept that experienced investigators do most of these, but we've made a comprehensive list of, I think, 32 different points to consider that hopefully will make it easier to set up sites by doing more preparation. So infrastructures are facilities and services that can be used multiple times. They can catalyze these discussions by giving you the tools that you need, rather than you having to find the tools repeatedly. So this is about avoiding reinventing the wheel. Infrastructures do need resources, so you need to include them in your grant applications, but they give you a broader range of expertise than you've had before, and it saves writing all these operating procedures every time you do another study. So if we map some research infrastructures onto our study plan, the first one I draw your attention to is the European Pediatric Translational Research Infrastructure, EPTRI. This is under development, and the idea is to bring together everybody who does work before a clinical trial is needed. So the animal work, the pharmacology work, the formulations work, the biomarker development work. At the moment, you have to come to meetings like this and randomly meet people over coffee or over beer or whatever you do to meet people. And the idea is to have a system of everybody who's interested in pediatric research with keywords for their interests, so that you can say, I want to study connexin 2 in 22 weekers on Tuesdays, and then people can give you a list of people who've got the methods to do that, who've done it before statistically, and so on. So we're trying to systematize the development of collaborations. And this is before the trial gets designed, all the information you need to design a good trial. The next group that you could work with is a European translational research infrastructure, and EATRIS brings people together and helps move down the translational research pipeline. So if it's about IP, it's about protecting your ideas, it's about bringing in the right people so that you're one step ahead of regulatory or financial requirements. So they're very efficient, but quite expensive. Another group to work with is the European Clinical Research Infrastructure Network, ECRA. They have national correspondents in a number of countries, and what they do is help move a protocol from one country into multiple countries. So you're in northern Norway and you want to do a study in Poland and France and Portugal, and it's very difficult if you're in northern Norway or northern England to find the right people. So rather than come to conferences and spend 3 years working out who to talk to, you can work with ECRA, and they'll put you in touch with ethics committees, they'll put you in touch with statisticians and trial managers in each country. So a very useful resource for moving academic studies between countries. And here are some contact details for these organisations, and I'd recommend them to you as good ways to speed up the setup of research. Okay, so coming on to C4C, Connect for Children, we're planning to contribute to this pipeline in two main ways. First of all, by giving expert advice and building collaborations at the start of this process during the gaps and ideas and discussion, bringing in experts in extrapolation and modelling and simulation and measurement and natural history, all those things that the gaps that we need to pay attention to, and also providing a sounding board of other neonatal experts to help people clarify their ideas. And we're very lucky that the leader of the neonatal expert advice group is in the room, Professor Alakat, if you contact him. So we have a college of 300 experts in every bit of paediatrics, from neonates through to adolescent and mental health, all the methodologies, who, when the system is up and running, will be able to give advice. They'll expect something in return. You'll have to pay them, or you'll have to put them on your papers, but it's having a system to get in touch with people rapidly in a reliable way for industry and for academia. The other group we're setting up is a clinical research network, and this is designed to make life easier, not just moving protocols from one country to another, but doing trials in sites and trying to simplify the process of contracting and ethics and improving the quality of work that people do in sites. So this is the second part of Connect for Children. So C4C is funded by the Innovative Medicines Initiative. It's a public-private partnership including 10 large pharma companies and 19 national hubs and a number of other organisations, including EFCNI and other patient advocates. So we're looking for national hubs because we found in the UK, in Austria, in Finland, that by having a national hub you can speed up trial setup and you make it easier to do things. So we want to spread that idea across the rest of geographical Europe. We've mentioned the expert advisory groups. We're developing an educational program for health professionals and awareness raising campaigns. Why is research so important? So at the moment we have a paediatric good clinical practice course that we're going to roll out, and then we're also building data standards so that when you talk about gestational age you mean the same thing in all the different trials, so we can make comparisons and data reuse easily. So currently, 18 months into the project, we've got 19 hubs operational, the expert groups are working, the educational portal is working, the metrics for how to judge how we do are on co-production with industry and academia, as is the business case. And we're working with the International Neonatal Consortium on data standards to make sure that people have access to consistent, controlled data terminologies. We're working on 8 proof of viability studies. There are 4 academic studies that have been funded, one of which is neonatal, called Tria Kappa, looking at paracetamol to prevent patent ductus and improve outcome. And what we're doing is setting up consistent systems so that people can produce reliable, consistent work more quickly than they are before, and trying to take a national step on this so that you don't have to go through multiple lawyers every time you try and set up a study. And this cartoon here is about the different modules of the C4C support system, working our way through different steps so that people have consistent ideas about budgeting and contracting. And if we can remove 1 or 2 months from each of these steps, then we'll make a big difference to the efficiency of the clinical trial system across Europe. So these are the members at the moment. The blue ones are the sites that the countries that are working with. For these purposes, Iceland is is working with Finland, and we're currently talking to Denmark, Lithuania, and Latvia about expanding into those countries. I'm going to visit Turkey in a few weeks' time, and we're just looking to expand as time goes by. We have these hubs. We have a number of specialty groups, the HIV, the rheumatologists, the cancer, the cystic fibrosis groups, and working with many children's hospitals. We'll have 166 sites in the proof of viability studies, and the idea is we use those 8 studies to learn how to work together and then scale up the activities within 4 or 5 years. I mentioned global networks. We're working with 2 partner networks in the US, IAC, which is Institute for Advanced Clinical Trials for Children, and also a global network from Duke. Of course, the global network from Duke can only involve Americans, but that's what happens when you have a World Series or something global in the US. We're working with Canadians, working with Japanese networks and the Pediatric Trials Network of Australia to try and improve the way that we work across jurisdictions, because neonatal drug development has to be global, and we have to be aligned about the design and implementation of studies. So this is how we're operationally putting together. C4C has a single contact point that all these different groups can come to, including the European reference networks. We have direct links with national hubs and sites, but we're working very closely with the specialty networks, working closely with these other infrastructures to make sure that we have consistent offers, and the expert advice groups as well. So the key thing is a single point of contact that will then efficiently pass information to the right people. So we're learning how to work. We're taking baby steps. We have to learn to tie our shoelaces before we can start to run. So please be patient, but over the next 3 or 4 years we will scale up, and we aim to be open to all academic and industry sponsors within 3 or 4 years, covering drugs, devices, immunizations, biologics, all sorts of things across geographical Europe, and all types of medicine studies. So this is the team. So communication that connects with children is the key email, and I'll summarize by suggesting that the way to set up a site for a clinical trial is to design the protocol well, to start with existing information, and then aim to make a single well-defined contribution to the literature that will be impactful, and then match design, rigor and stringency to the aim. It's no good having 100% source data verification if you're not going to use that for regulatory purposes. Prepare thoroughly, and please comment on the guidance about how we can prepare, and then collaborate. Please drink beer with your colleagues, but also please use research infrastructures to speed up your development of clinical trials. Infrastructures will facilitate your work, but they won't do all the work for you, and research infrastructures will not provide a recipe. It's a little bit like using a ventilator. I don't have a recipe for ventilators, but I know a little bit of physiology, and so if I want to increase the mean airway pressure, I can do this and this and this, except on Tuesdays or when my colleague's on call, when I do that, that and that. And research infrastructure will give you that kind of advice. They're not going to say, put this setting here, put that setting there, and everything's done. They'll think about them as a physiology rather than as a way of doing your work. So I'll pause there, stop there. Thank you very much.